I'm Alessandro Marcello. I'm the group leader of the Laboratory of Molecular Virology of the ICGEB, and we basically work on viruses of human interest. BDU for us is very important because several of the reagents that they are preparing, such as for example as interferon, we use it in our study. And more recently with the COVID epidemic, we needed to produce tools for diagnostics such as the recombinant spike protein, and with them we could develop the purification protocols for this protein. Also, we purified a recombinant antibody against the virus, which is very useful for our research because it allows us to track the virus's infected cells. Clearly, this platform is really instrumental also in infectious diseases because the production of neutralizing recombinant antibodies is one of the approaches for the treatment of this disease. In our lab, we study the interaction of viruses with the host. In particular, we are interested in viruses that infect humans, and in the pre-COVID era, we were mostly interested in tropical diseases, including viruses of global importance like dengue virus, Zika virus, and other viruses like chikungunya virus that impact not only the tropical areas but also Europe and Southern Europe. In addition to this, which is more basic, we have interest also in translational aspect of research. In particular, we are working on virus surveillance and diagnostics by collaborating, for example, on a network of European countries for the surveillance of certain flaviviruses which are delivered by ticks, as well as developing diagnostic tools, point-of-care diagnostic tools, portable diagnostic tools with companies such as Halifax that are interested in diagnostic of viral diseases. So clearly last year in March, when we had the first cases here in the Regione Friuli-Venezia Giulia, we immediately started our interest into SARS-CoV-2, which is the virus causing COVID-19 disease. We isolated the virus from infected patients in the region. This means that we could grow the virus in cells in our laboratories because we have a facility for the manipulation of pathogenic diseases, biosafety level 3 laboratory. Isolation of the virus allows us to study the virus, so the life cycle of the virus, as well as allows us to find antivirals that are specific for this viral disease. In addition to this, we sequenced together with institutions in the region, such as Area Science Park and the Azienda Ospedaliera della Azugi. This is very important to understand how the virus evolved during the epidemic, starting from March last year up to now. So now we are experiencing these variants of the virus, and we are currently sequencing what is circulating in our city and our region. In addition, support to the industry, because being able to manipulate the virus and being able to measure infectivity of the virus allows us to collaborate with several industries, not only in the developing of antiviral molecules for treatment, but also for tools for disinfection, such as physical and chemical tools. I'm talking about UV light or detergents that can be used to inactivate the virus on various surfaces, including a collaboration with Fincantieri, who has a real need to have a safe air conditioning system in their cruise ships and cabins. And so we are collaborating on developing these tools. This is the translational aspect of what we do. So we are both a laboratory interested in basic research and translational research. Translational research means translate the findings that we get in our current basic research into practice, into something that can be delivered to the patients or delivered to the society. And the BDU is at this, establish this link, allows us to go from the laboratory level, which means something that we can do at the laboratory level, so with our hands in the lab, to a more industry-like production in a GMP facility with all the regulation for the manufacturing that are followed carefully. So this is very critical for us to be able to translate what we find in our research into a product.
I'm Serena Zacchigna. I'm the group leader of the Cardiovascular Biology laboratory here at the ICGEB. I'm a medical doctor, and since the time I got my medical degree, I've been always involved in research with a particular interest in developing novel therapies for cardiovascular disorders. Our major interest now is understanding why the heart is rarely affected by cancer and trying to understand to what extent this is linked to the lack of regenerative capacity of the heart and the incapacity to form new blood vessels. So this research, of course, is an ambitious topic aiming at developing novel therapeutic approaches for both cancer and cardiovascular disorders. We always use recombinant proteins in our daily work. Recombinant proteins are precious tools for research, but also can work as biotherapeutics for human diseases. For instance, we are currently collaborating with the BDU in producing a protein that have the potential to counteract the invasive properties of cancer cells. Of course, industries are very well interested in some facilities that we have here at the ICGEB, for instance, the high throughput screening facility, and more particularly related to our work. I'm a medical doctor, so my research is always linked to the clinics. We have specific skills in the development of animal models of human diseases, and also we work with primary cells and tissues from patients. We collaborate with Bayer for the development of novel therapies for uterine fibroids. We have a collaboration with the local industry producing bioreactors to culture human cells. And we have a long-lasting collaboration with a company producing lasers. We already started with some skills that were precious because in the past we learned how to culture human lung cells and to culture human tissues, and how to analyze histologically the lungs. So these skills were precious, and we exploited this knowledge to investigate what happens in the lung of people who suffer from COVID-19. And we are developing both cellular models to study the disease in-house. Together with the company producing laser, we also develop an aerosol system to mimic the natural infection and have simpler models where to study the mechanism of infection, and also developing novel therapeutic approaches. Again, we were interested before in pulmonary fibrosis, and now fibrosis is expected to become one of the major long-term consequences of COVID-19. So the development of novel therapies able to counteract pulmonary fibrosis is expected to have a huge impact on our capacity to help people who had suffered from COVID-19 and will have to face the long-term consequences, the so-called long COVID.